As we already discussed in the previous lectures, plant remains are generally grouped by size. While macro remains are usually larger than 0.1 mm, micro remains are smaller than 0.1 mm. The most important plant micro remains are pollen, spores, starch, and phytoliths. The master program here offers some special courses on palynology and phytolith studies, so I will only shortly mention some basics in this presentation here. Phytoliths can be shortly described as ovule that formed intra- and intercellularly in plant organs. Techniques used to determine the different shapes are light and electron microscopy. Phytoliths are formed from silica that is in solution in groundwater and originally derives from weathering of silicate minerals in metamorphic rocks. In solution, silica exists as monosilicic acid. It is carried upward in the vascular system of plants and becomes concentrated during transpiration around the leaf stomata. The supersaturated solution begins to polymerize, solidifies, and forms solid opaline silica bodies within and between some of the plant cells. Grasses are the most widely distributed plants on Earth and produce many and varied phytoliths, which may be the reason why they are the most intensively studied plant family. More recently, phytoliths have been extracted and described from many other species worldwide. Phytoliths are found in nearly all plant structures, stems, leaves, roots, and inflorescences. In general, more phytoliths form in the aerial tissue than in subterranean parts. The formation of phytoliths in monocotyledons, that is, mostly the grasses, often involves the filling of a cell interior. Cell interiors act as molds producing solid phytoliths of different but repeatable three-dimensional shapes. Solid phytoliths are extremely resistant to decomposition and are therefore more frequently recovered from soils and sediments than those produced by woody species. Here are some examples of such solid three-dimensional phytoliths. This and the following slide provide a rough scheme of the extraction methods for phytoliths from modern plants and from sediments. The functional significance of silica is not obvious, and to some extent the mechanism of phytolith formation appears passive and may represent waste products. Alternatively, phytoliths provide mechanical support, preventing the collapse of cell walls under tension during transpiration. Moreover, phytoliths provide plants with increased resistance to browsing by herbivores and insects and attack through pathogenic fungi. In spite of incomplete understanding, it does appear that silicon is an essential element in the development of some species. Measurable amounts of microcrystalline structures have been reported within phytoliths, creating the possibility of direct dating using OSL methods. Phytoliths can contain inclusions of chemical elements such as aluminium, iron, phosphorus, copper, nitrogen, carbon, and others. Carbon occluded in phytoliths is the result of the trapping of plant cellular material during phytolith formation within living cells. It has been demonstrated that more than 50% of this encapsulated carbon is protected from oxidation and thus provides another source of paleo-environmental and paleoclimatic information. The development of EMS radiocarbon techniques using very small samples permits the dating of phytolith-occluded carbon. Moreover, stable carbon isotope analysis can also be used to provide additional information about the photosynthetic pathway of plants, that is, whether C3 or C4 grasses dominated a given environment in the past. Phytolith-occluded carbon has also been used to generate a late Quaternary isotopic record of atmospheric carbon dioxide. Phytolith analysis is a developing discipline, and there are still identification problems to be resolved. These are of multiplicity compared to seed remains, because many different phytoliths can be found in a single taxon, and at the same time one form can occur in many different taxa. A number of independent classification schemes of phytoliths so far existed, generally for specific geographic regions. There was a certain amount of overlap between these schemes, and over the years a number of attempts have been made to develop a universally accepted standard classification. Morphological differences in phytolith assemblages are often used to indicate changes in the relative abundance of forest versus grass cover. In case of correlation between tree-shrub phytolith fluctuations and oxygen isotopes from paleoclimate proxy archives, changes in the ratio of arboreal to non-arboreal phytoliths are interpreted to directly result from climate change. A number of such studies are available for different regions of the world. From the 1990s onwards, quantitative methods were put forward in phytolith analysis. Some researchers use multivariate statistics to distinguish between phytoliths from a large number of species. As already mentioned, stable isotope analysis of phytoliths can contribute considerably to paleo-environmental and paleoclimate research. Stable and unstable carbon occluded within phytoliths is protected by the non-porous glass-like microstructure of the silica body. Oxygen information about climatic conditions during plant growth is also preserved in phytoliths, providing information on paleoclimate change. The lower left figure shows an example of the mentioned stable carbon isotope analysis based on phytoliths. As a result, we see the range, variability, and changes of C3 and C4 grasses of past ecosystems. Most phytolith studies, however, focus on human and cultural development and are directly generated within archaeological research. Phytoliths on artifacts or dental calculus provide evidence of early human subsistence on plants. This is particularly relevant in areas with reduced preservation of carbonized plant remains, such as the tropics and subtropics. Phytoliths of progenitor species of modern crops are of interest in following up the roots of agriculture. Observed morphological differences between phytoliths found in wild and domesticated rice species have been used to document change from early exploitation of a wild resource to its subsequent cultivation. Others tried to differentiate phytoliths from maize and its assumed progenitor teosinte. In case of crop plants producing little or no seeds, such as banana, the analysis of phytoliths has proved useful in describing its early cultivation. Particularly in semi-arid to arid regions, irrigation was amongst the most important technologies with the intensification of agriculture and the emergence of early urban civilizations such as in southern Mesopotamia. According to Akkadian texts, complex relationships between natural flooding in early summer and agricultural production required the construction of canal systems in cooperation between several villages and their intensive management through a canal inspector. Despite some textual evidence, the dating of canal system is problematic, as their use may have continued into later periods. For this reason, we know only little about irrigation for many Near Eastern locations. The direct evidence of irrigation in plant remains would therefore help to solve this question. Some phytolith researchers observed that united cell structures in emmer wheat consist frequently of more than 10 cells if the crop has been irrigated, while those with only few united cells are more frequent in unirrigated cereals. This relates to the passive process of phytolith formation in cereals, which means that the amount of phytoliths directly depends on the SiO2 concentration in the water and on the rate of transpiration. This led to the hypothesis that the frequency of large united cell structures depends on the microclimatic environment. Experimental cultivation showed that the majority of phytoliths in cereals from rain-fed fields have 2 to 4 cells, while in irrigated crops more than 25% of the phytoliths had more than 10 cells. Chalcolithic emmer from Gilad in Israel showed a similar pattern like the irrigated modern emmer, thus also appears to have been irrigated. A major methodological problem with such an interpretation is, however, that possible different climate conditions have not been considered by the authors. Paleoclimate records, for example, indicate higher precipitation at the transition from the mid-late Chalcolithic around 6500 BP. Another aspect of interest is animal husbandry in relation to ancient landscape conditions. If dung pellets do not contain seeds, phytoliths preserved in the dung may inform on ancient habitats preferred for browsing animals. In general, grass phytoliths are dominant, but also other floristic elements can be present. Here an example from the Bronze Age site of Tel Mosan in Syria. The ancient samples seem to have the same composition as the modern. Both are dominated by grasses. The large number of phytoliths from culms and leaves suggests browsing on complete plants, instead of feeding cereal grain, as has occasionally been claimed by some archaeologists. Let's have a very short glimpse on palynology, which will be taught in the following semesters in extra courses. Pollen grains are unicellular microscopic organs that contain the male generation of the seed plants. Traditionally developed within historical geobotany, the study of fossil pollen and spores is the principal technique for long-term vegetation reconstructions. The first description of pollen is documented from the end of the 17th century, and the first pollen diagram has been published by Leonard von Post in 1916. Pollen stratigraphical results have greatly shaped the development of ideas on the nature of vegetation and climate during the glacial-interglacial cycles. They have provided unique insights into long-term ecological patterns and processes and the sensitivity and response mechanisms of plants and vegetation to climate change. With Franz Filbers since 1949, they have also shed light on the origin and spread of early agriculture and on human impact on the vegetation. Plants liberate enormous quantities of pollen into the air. There are a number of ways pollen is distributed, but it is most efficiently transported by wind. Those are therefore widely and evenly spread, while others are usually underrepresented. The spread of pollen by wind also results in methodological complexities, some of which constitute the most important limitations and pitfalls of the method. Fossil pollen records are available from various sedimentary environments. The pollen concentration of natural sediments is usually high. In lake sediment, often of a magnitude of 100,000 grains per cubic centimeter. The sedimentary pollen records are therefore unique in their stratigraphic detail. The following description of the historical overview on palynology has been taken from Richard Bradshaw's contribution in the Encyclopedia of Quaternary Sciences. Pollen analysis, or palynology, lies at the interface of geology and biology, but has close contacts with other disciplines such as archaeology. As with the macro remains, pollen analysis has tended to develop in different directions in different countries, largely dependent on the interests of the influential pioneers in the field. The study of quaternary vegetation history began in Scandinavia at the end of the 19th century, primarily through the work of Blitt and Cernander, botanists who examined peat bog stratigraphy and plant macrofossil content to develop a scheme of post-glacial climate change. Their ideas were further developed by the Swedish geologist von Post with the technique of statistical pollen analysis, that is the most widely used method for reconstructing quaternary vegetation history today. The pioneers of pollen analysis in Sweden, von Post, Nielsen, and Erdmann, were mostly attached to geology departments, and today most Swedish students of paleoecology are still trained as quaternary geologists. Bergs reviewed the interesting development of pollen analysis in Scandinavia. Erdmann, who worked in the shadow of von Post, was the first to publish widely on von Post's method outside Scandinavia, and paid an influential visit to Scotland, Ireland, and the Isles of Man in the mid-1920s. He inspired, among others, Godwin to work on the East Anglian fens and submerged forests of coastal peat beds. Godwin published a critical assessment of the potential of von Post's method and dominated the British paleoecological scene between 1940 and 1960, becoming professor of botany at Cambridge and founding a great British centre of botanical paleoecology. The history of the British flora, a factual basis for phytogeography, first published in 1956, is still a useful paleoecological text summarizing early pollen work in Britain. Godwin emphasized the close links between pollen and plant macrofossil studies. Pollen studies developed somewhat independently in Central and Eastern Europe, with Rudolf, Bertsch, and Filbers being important names among the pioneers. After the initial enthusiasm and excitement from the pioneer days of palynology, the next significant development was the routine application of absolute chronologies to late quaternary sedimentary sequences through the development of radiocarbon dating. This development took place during the 1960s in research groups with access to sufficient funding and contributed to a generally increased level of precision in the construction of vegetation. Quaternary pollen analysis developed from being merely a method of geological correlation into a useful tool for exploring vegetation dynamics. Davis, Huntley, and Burks published continental scale maps showing the Holocene spreading of tree taxa from their glacial refugia to their present distributional limits. Their work illustrated the dynamic nature of tree distributions that have undergone continuous change. The subsequent work of the Cooperative Holocene Mapping Project, the so-called COMAP group, demonstrated that vegetation was continuously changing in response to climate forcing, bringing into focus the quasi-equilibrium situation between vegetation and climate. This line of research, which originated in a strictly geological approach, has led to a paradigm shift in biology. The concepts of climax, vegetation, and traditional succession were shown to be largely untenable under typical disturbance regimes with a constantly changing climate. This development in the subject is a good example of results from one discipline leading to constructive development in another, the best possible outcome of interdisciplinary research. The increasing quantitative and spatial precision in vegetation reconstruction has led to exciting new insights being generated in the research areas of evolution, ecology, and conservation biology. These topics included plant migration, disturbance biology, population ecology, and the long-term evolution of genetic structure. Great progress has also been made in placing archaeological sites in a broader environmental setting, and all these research topics cut across the disciplines of earth and biological sciences and archaeology. Most approaches to reconstruct past human impact from pollen data are based on indicator species. As we remember from the previous lectures, most modern plant species have specific ecological requirements, thus can be used in uniformitarian argumentation to indicate specific ecological conditions in the past. With the indicator approach, the presence of a few pollen grains from an indicator type is the basis for interpretation. The approach provides a way to identify variations in land use and landscape management through time. It also makes it possible to describe relative changes in the extent of total human impact and different management types. The classical indicators for cultivation are pollen of cereals and related weeds. Finally, you can have a look on how pollen and macro remains complement each other in and around archaeological sites.